mRNAs or selectively degrade only certain mRNAs. And so that's a form of differential gene expression. And just to illustrate this, let's say this particular mRNA has a sequence on it, GU, CU, that's unique to that particular mRNA. And so if the cell wants to downregulate just this particular mRNA, it has to somehow key in on this particular sequence on it. And here's the way it works. The cell can manufacture some small RNA molecules that are sometimes called miRNAs, stands for microRNAs, because they're much smaller than the messenger RNA itself. Or sometimes they're called siRNAs, which stands for small interfering RNAs. And these miRNAs or siRNAs, whatever we want to call them, are complementary in sequence to the sequence of the messenger RNA that the cell is trying to downregulate. And remember, there's this almost magical property of nucleic acids. Whenever you have two complementary nucleic acids, they can find each other. That almost like magnetism, they're going to want to come together. And so these small interfering RNAs, these si or miRNAs, will come in magically. Well, I shouldn't say magically; it's hydrogen bonding. But they'll find just this particular mRNA because they're complementary to that sequence and bind to it. And what they found is that the cell does manufacture does indeed manufacture these miRNAs, but the miRNAs get hooked up to these blocking and RNA degrading enzymes first. And so in other words, the miRNA drags the blocking or degrading enzymes just to the particular RNA, just to the particular RNA that it's complementary to. And so the cell that way can selectively block or degrade just certain mRNAs. So to illustrate here, if the cell wants to not express this particular mRNA, it has an miRNA that's complementary in sequence to it, and that miRNA first has become attached to either a blocking protein or a degrading enzyme. And so, these two molecules work as a team. The RNA part right here, the miRNA, is responsible for binding only to the messenger RNA that's supposed to be inhibited, and the protein is the one that's either going to block the ribosome or degrade the messenger RNA. So when it binds on like this, well, the ribosome is either blocked or the mRNA is degraded, whichever type of protein that the miRNAs become hooked up to. You buy it? Okay. There's that concept is called RNA interference, where the cell manufactures these miRNAs to stick to some of its messenger RNAs, and the miRNAs become bound to the blocking proteins or the RNA degrading proteins. And this RNA interference is a fairly new concept in biology. It's only a few decades old. Before that, they had no idea that cells could do differential gene expression that way. And I want to tell you a little bit about where these miRNAs come from. And I guess I want to start by telling you where they don't come from. And I hate to lecture on incorrect things that aren't true, but it kind of tells an interesting story. But anyway, from what I'm about to show you here is not how this works. Here's my Buster symbol. Let me show you how they thought it worked at first. Concentrate on the messenger RNA in red there for a moment. If that messenger RNA includes the sequence GU-CU, let me put a few things on there. How about a UCC GU-CU? AA like that. But that's the messenger RNA. and if we look for the gene that made that messenger RNA, do we find the GU-CU in the gene's template strand or non-template strand? Who recalls? Template or non-template strand? Come on, somebody be brave. It's in a non-template strand. Because here's the gene now. The messenger RNA gets made by RNA polymerase II enzyme binding to the template strand and making a complementary messenger RNA to it. So this would be, I have to think, this would be A, G, G, C, A, G, A, T, T is what the template strand would look like. And the non-template strand, remember, is the one that looks like the messenger RNA. The non-template strand would be T, C, C, G, T, C, T, A, A. Sound okay? Correct so far? All right, so the message here is that the mRNA looks like the template strand, but it's complementary to, the mRNA looks like the non-template strand, but the mRNA is actually complementary in sequence to the template strand. Now, when they first learned about these interfering RNAs, these miRNAs, they knew that they were complementary to the messenger RNA. And so if we wanted to make a complementary RNA, which strand should be transcribed? Template or non-template? No. Not template. Because in other words, if we wanted to make something that was CA-GA, which strand do we transcribe? The non-template strand. Imagine RNA polymerase bound onto the non-template strand, which again, it normally doesn't do. RNA polymerase bind onto the template strand. But if RNA polymerase bound onto the non-template strand, look what you'd get. You would get UUAGACGGA. And so notice you get a CAGA as part of that sequence. And so people for a long time thought, or at least initially they thought, well, that's where the interfering RNAs come from. You see, they said the RNA polymerase must somehow bind to the non-template strand and transcribe that, and that would give you a sequence that's complementary to the sequences in the actual messenger RNA. And so it was a brilliant idea on somebody's part, but it just turned out not to be true. They looked at it, and it turned out not to be the case. So where do these complementary miRNAs come from, if not from the non-template strand? Well, oh yeah, so there it is, degrading it. There we go. So anyway, the interfering RNAs, the miRNAs, don't come from the same gene as the actual mRNA. They come from an entirely separate gene. The cell has genes whose entire function is just to make interfering RNAs that are complementary to the messenger RNAs, which is almost a mind-boggling concept, because we have 25,000 genes, and that could potentially mean you have 25,000 other genes which just make complementary interfering RNAs to the messenger RNAs coming from the original set of 25,000 genes. Now, I don't know if all of your 25,000 genes has sort of a matching gene that makes interfering RNAs to that gene. Matter of fact, I doubt they all do. But the point is at least some of your 25,000 genes do. So at least some of your 25,000 genes, there's a kind of a matching evil twin gene, so to speak, that makes complementary RNAs to it so that your cells can do RNA interference to those mRNAs.
Is there some type of, how does the cell know what mRNAs are split around in this cytoplasm at any given time?
At any given, well, I guess you might say the cell knows which RNAs it's transcribed. And so I guess I would say if the cell has an activator protein that allows it to make this particular messenger RNA from a gene, maybe that same activator protein activates the genes to make the interfering mRNAs for that. That way, for any gene it expresses, it can also express some interfering RNAs for it. All right, so anyway, the point here is these interfering RNAs come from an entirely separate gene whose whole purpose, so to speak, is to make these interfering RNAs to decrease the expression of this original mRNA over here. And so what biologists did is they started hunting around for the genes that make the interfering RNAs, the miRNAs. And they found those genes. And when they looked at the product of those genes, the genes that make the miRNAs, what they found is this. The original RNA that comes out of these interfering genes is a long RNA that folds back on itself by complementary base pairing. And so regions of this mRNA, well, I shouldn't use the word mRNA, regions of the interfering RNA are complementary towards each other, and so you get some double-stranded regions there. And that's kind of unusual because for the most part, RNAs are single-stranded molecules. It was DNA that's supposed to be the double-stranded nucleic acid. And you know, this is a little bit different because when DNA is double-stranded, it's two separate strands, like the template strand and the non-template strand, are two entirely different molecules that come together by complementary base pairing. Here we have only one RNA molecule, but parts of it are complementary to itself, and so it does fold into some double-stranded region like that. Anyway, these interfering RNAs first get produced as a long RNA that has some self-complementarity like this and folds back on itself. And then what they found is there's a protein in the cytoplasm called dicer, in the sense like you dice something when you chop it up. And this dicer protein then comes along and finds this double-stranded RNA and dices it up. And that's where the small little interfering RNAs come from chopping up this big thing right here.
Are the genes that express the microRNAs?
Yeah, these guys, oh yeah, the gene.
Would they be found in the same genes for which they're expressing the microRNAs? I'm saying if there's a gene for some specific thing, that the genes for that are encoded in mRNA, are you going to find the same gene for the microRNA on that same chromosome?
That's a good question. So the question is, if there's a gene for protein X, do I find the interfering RNA gene on the same chromosome? That's a great question. I don't know, but just if I had to guess, I'd say not, because even genes that function together, where the gene products function together, are often on entirely different chromosomes. So I suspect not is the answer. Anyway, so as I said, this is a relatively new field area in biology, this RNA interference. And people kind of wonder, well, this seems so bizarre, where did it come from? And the leading hypothesis is this, that normally we don't get double-stranded RNA inside the cell because RNA is usually single-stranded. DNA is double-stranded, but not RNA. But one type of organism where you do find double-stranded RNA is in viruses. Well, that's not what I wanted there. Certain viruses, matter of fact, a lot of viruses are RNA viruses, where the viral chromosome is RNA. And for some of these RNA viruses, the RNA is actually double-stranded. And so what they think this started out as is a viral defense system, that the cell said, anytime I see double-stranded RNA in my cytoplasm, I'm going to assume that that's a viral chromosome. And so the cells evolved a protein to chop up those viral chromosomes. And somewhere along the evolutionary line, the cell said, hey, these little bits of RNA that I end up with can be useful because I can use them to help regulate my messenger RNAs if they're complementary to my messenger RNAs. So the cell evolved this RNA, this defense system against RNA viruses, and it eventually evolved into RNA interference for controlling the expression of its own mRNAs. At least that's the leading theory of where it came from. There it is, yeah, so the double-stranded mRNA is virus-like. You normally find double-stranded RNA only in viruses. All right. Any questions on RNA interference?
Will we be tested on this specific piece?